Thank you, Dr. Warren. As mentioned, my name is Caitlin Tydings, and I'm a pediatric oncologist and medical officer within the sarcoma team in the Division of Oncology 3. Today, I'll be providing general clinical considerations for first-in-human trials. We will start briefly going over some general good practices, followed by discussing important components of new INDs, common mistakes we see when reviewing new INDs, and we will finish with resources you may find helpful. Here are a few good practices that one should be aware of when developing a first-in-human trial. It is very important to engage a trialist or trialists who have expertise, and this is ideally in both the disease indication as well as with first-in-human trials. Next, pre-INDs can be requested to discuss if you have enough of the package to submit an IND, and this may help streamline the IND review process. Throughout the development program, it will be important to distinguish drug activity, which reflects the biologic effect of the drug, from clinical benefit, which reflects clinical effect that is meaningful for a patient, and failure to do so may waste resources. With that in mind, we will jump into an overview of INDs. As many of you know, there are three major components of the IND: chemistry, manufacturing, and controls, or CMC, preclinical data and analysis, as well as the protocol itself. The review team at FDA is multidisciplinary. Regulatory project managers facilitate all communications between the sponsor and FDA, as well as between the FDA reviewers themselves. Our chemists review the CMC portion, toxicologists review the preclinical data, and pharmacologists, clinicians, and statisticians review the protocol. FDA teams discuss new INDs at weekly safety meetings, and we send information requests or IRs to the sponsor for revisions or clarifications. These can include potential hold comments, which are changes that must be made by the sponsor and pertain to safety, or they could be non-potential hold comments, which are suggestions by the FDA that the sponsor should think about. Following these communications, the study may be placed on clinical hold, or the study will be declared safe to proceed. For first-in-human and exploratory or proof-of-concept trials, FDA focuses the review on safety. If we determine a new IND to be safe to proceed, it may be with or without contingencies, and these are usually discussed during the back and forth of IRs. For example, the trial may be contingent on uploading a revised informed consent document. If we determine a new IND is not safe to proceed, the IND is placed on hold. Partial holds include allowing the trial to proceed up to a certain part. So for example, up to a certain dose level because there was insufficient safety data to allow escalation beyond that, after which the sponsor must discuss with FDA and address FDA's concerns before continuing. Complete holds require that sponsors address all potential hold issues before the trial can begin. These are the various components of the IND we will briefly go over. For dose escalation, one can follow a rule-based design such as a 3+3 and its variants, accelerated titration design, etc. These are often easier to implement. For example, 3+3 is very commonly used, but they may not be the best at determining the maximum tolerated dose and the recommended phase 2 dose, and more patients may be treated at a subtherapeutic dose. Model-based designs use statistical models, including continuous reassessment model, CRM, modified CRM, Bayesian optimal design, etc. These are better at determining the MTD and RP2D, but are more challenging to implement and require biostatistical support for decision-making. Dose escalation may be more rapid, which risks them being too aggressive, and if there's no overdose control, you may treat more patients at a toxic dose. It is important to talk to your statistical colleagues about the best design to use and what your institutions and programs can accommodate. The goal of dose escalation is determining the RP2D, the MTD, or if the MTD is not achieved, the maximum administered dose, or MAD. The RP2D is the dosing regimen that you decide should be further studied based on clinical PK and PD, safety and tolerability of the dose, and preliminary efficacy if you looked at that. One thing to consider is if the drug is being taken orally or is going to be taken daily or frequently, long-term tolerance and toxicity should also be taken into account. If you're using the 3+3 design, the MTD is defined as the dose at which 0 out of 3 patients experience a dose-limiting toxicity, or 0 to 1 out of 6 patients experience a toxicity. Other designs have other definitions. Generally, we want the highest probability of risk of drug toxicity to be less than 30%, and the target risk of drug toxicity should be aimed at about 25%. But there is some flexibility depending on the design, so we will provide feedback if needed during our review of the protocol. Acceleration of development beyond dose escalation can take traditional approaches such as activity estimation designs. These can be single-armed or randomized. For single-arm, use a design that would limit exposure of a large number of patients to ineffective drugs such as a Simon 2-stage. And for randomized designs, use a common control arm and avoid formal comparison between experimental drugs. Some newer strategies include master protocols, including basket trials, which looks at multiple cancer types with a common genetic mutation like NTRK, or umbrella trials, which looks at a single cancer type but evaluates multiple drugs. You can refer to the guidance at the bottom of the slide regarding enrolling patients for clinical trials investigating drugs in the non-curative setting and what you can include in the informed consent document regarding available therapy outside the trial. We have done a lot of work to expand eligibility criteria and allow patients the option to receive an experimental drug. The focus has been on later phase trials, but even first-in-human trials could benefit from careful consideration of eligibility criteria to improve patient access to trials. For dose selection, the dose range is guided by non-clinical toxicology, and dose escalation can be linear or logarithmic. Generally, linear increases are used for small molecules with up to 100% increases, and logarithmic increases are used for proteins. If we expect a fair amount of toxicity, we will recommend conservative dose increases. FDA is working hard to find ways to better optimize dose selection, and we have a working group called Project Optimus undertaking this very important task. Dose-limiting toxicities should be included in the protocol by way of defining the observation period, which should be at least 1 month, defining what a DLT evaluable patient is, with some example language here, and listing the DLT criteria itself. This is where it can also be helpful to have a pre-IND meeting, as there's often some back and forth regarding DLT criteria. Dose modifications must be included and based on anticipated toxicities and/or the dose-limiting toxicity. You can also consider dose reducing to the previous tested levels in the trial. Just a quick note for safety monitoring, you must monitor and collect all adverse events during first-in-human trials. This is for up to 30 days from the last dose if a drug is not immunotherapeutic, and up to 90 days from the last dose if the drug is an immunotherapeutic, and if delayed onset of adverse events is possible. The parameters to monitor should be based on what's known about the drug, the anticipated toxicities, and the mechanism of action, and should be a minimum of a CBC and CMP and more specific testing based on what I mentioned. Both subject stopping and trial stopping criteria must be included in the protocol. I will quickly note that terminating the trial for safety is usually a decision point that is already part of the dose escalation, but bear in mind that the MTD may not be reached depending on the toxicity of the drug. Finally, I put a short list of some common pitfalls I see when reviewing INDs. This is not exhaustive. For example, some protocols we see state that they collect AEs related to the study drug. However, all AEs should be collected regardless of relatedness, as all AEs are considered related to the study drug in first-in-human studies. We often make notes of aggressive dose escalation and not starting at least dose -1 for combination regimens. I've included some helpful resources that you can refer back to for FDA websites, guidances, and where to find templates. And I want to thank you all for your time today. We will next be moving to to the Q&A portion of this session. Thank you.